Today we're going to start the AP Biology Diffusion and Osmosis lab, where we're looking at diffusion across a plant cell membrane, or osmosis across a plant cell membrane. And in this lab you typically work with potatoes, but I always give my students an option of choosing potatoes or carrots or sweet potatoes. If you are choosing a potato, then I recommend using a French fry cutter to cut the pieces so you get equal pieces. To keep things a little simple, what I'm going to do today is I'm going to use carrots to collect our data. And baby carrots work great. I just recommend to my students that they cut it in half lengthwise. And so I'm going to do that for one of them. And then what you really want to do is you want to rinse them, dry them, and weigh them. And you're going to set up 6 cups. Each cup will have 6 pieces of the carrot, so 6 halves of the carrot. And we're going to mask them today. We're going to add each of the different colored solutions. And what we're trying to figure out is which molarity goes with each of the colors. So we know that we have a 0 molar, 0.2, 0.4, 0.6, 0.8, and 1 molar sucrose solutions. We just don't know which one is which. And so we're using the cells, the carrots, potatoes, sweet potatoes, those cells to figure out if we can determine the molarity. So what I'm going to do, I'm going to start with these. I'm going to just rinse them off real quick. And I'm going to take some paper towel and dry them off. So just a quick patting down for the initial mass of our carrots. So I'm going to put my little cup on the scale. I'm going to zero it out, and then I'm going to put my 6 carrot pieces in there, and I'm going to write with a Sharpie directly on the cup 26.52 grams. So 26.52 grams. So that's my first cup. And then I've already cut a whole bunch of carrots. So I'm just going to rinse them off real quick. So I don't have to cut them all. And then we're going to dry them. And then each cup is going to get 6 pieces, and we're going to mass each of those. So we'll take 2, 4, 6 pieces, pat them dry, put them in the cup, and then again we're going to write that on the cup. So 27.26. 27.28, and then I'm going to do another 6 pieces. That's 30.15. 22.13, 24.28, 24.83. And so now we have filled all of our cups and masked them. So those are going to be our initial masses. And then what we need to do is we're going to add the different colored solutions to each of these cups. One of them will get the red. One of them get the orange. And gets the yellow. Green. Blue. And finally purple. Okay, so now we've got our 6 colored solutions and our initial masses on each of those. And so what we're going to do is we're going to cover them with plastic wrap and we're going to let them sit for 24 hours. Okay, I've put them into a tray and covered them with plastic wrap, so they're going to sit there for 24 hours to 48 hours, and then we'll take them out and rinse them, dry them, and weigh them again to get our final mass, and then we'll see if the mass increases or decreases, and we're going to use that information to see if we can determine the molar concentration of each of those 6 sucrose solutions. Our samples have been sitting for about 24 hours, so at this point what we need to do is remove the carrots from each of the different liquids. We're going to just gently dry them off and we're going to weigh them. Our samples. And then we have to also record, so we've got our initial mass recorded. So we're going to zero balance it and we're going to remove our carrots. Just going to get the excess water off of them. And then I'm going to mass them. So that is 25.92 grams for the red one. We've got our orange. And that is 23.39. And then yellow. And that one 31.74. We've got green. That one is 17.82. We've got blue. and 18.83. And finally, the purple. So 24.86. Okay, now that we have all of our data collection, what we're going to do is we're going to calculate the mass difference first of all, and then we're going to calculate the percent change in mass by taking the final mass minus the initial mass divided by the initial mass times 100. And once we have our percent change in mass, then we can compare them to see which solutions or which caret cells increase the most in mass and decrease the most in mass. And then we can use that information to determine the molarity of each of the different colored solutions. So we need to do a little bit of math now.